This video is cell cycle M phase. I want to point out that some of these terms should be pretty familiar to you already from other classes, including chromosome and chromatin. I'm not going to talk about them a whole lot, but you might want to look them up in the index of your textbook for cell and just kind of refresh your memory about them. So the first question here is, what's the central problem, and in quotes problem, for a cell that's in the M phase? Also a reminder that M phase includes both mitosis and cytokinesis. The M does not stand for mitosis solely, nor does it stand for metaphase. And so the deal is that for the cell, this kind of problem, or more so a task or challenge, is in this answer here to divide up the chromosomes so each cell has one copy of each chromosome, not two copies of one chromosome and missing a copy of another chromosome. So there's a lot of coordination that has to take place here. Basically, this coordination is done by two cytoskeletal machines, one of which is the mitotic spindle, the other of which is the contractile ring. The mitotic spindle is shown in this cell, which is completing mitosis as well as initiating cytokinesis, and basically the mitotic spindle is shown in green. So that's especially the dark green structure here. And my question for you is, which parts of the cytoskeleton or part of the cytoskeleton is used for each of these machines? Secondly, I'll just point out what the contractile ring is. So that's shown in red. So that's this stuff going on here around the constriction, which is basically where the cell will eventually continue to cinch in and eventually divide in two along the location where that contractile ring is. So as you may recall, the microtubules are typically shown in green, and that is what makes up the mitotic spindle. And then for the contractile ring, that is primarily made of actin. Also, it uses some myosin motor proteins to be able to do these constrictions. It really is a challenge to coordinate both of these events at the same time. Again, the mitosis part is dividing up the nuclear material, the chromosomes shown here in brown, and then cytokinesis is cleaving the cytoplasm of the cell in two. The cell needs a very organized and methodical way to divide up this material, for example, to make sure that this chromosome here is separated from its identical copy or sister chromatid that's over here. And likewise for this one, its sister chromatid, this one here. They all kind of look like they're identical to each other, but really the one that's right across from it is the only one that it's identical to. And so our big goal is to understand how this happens. Basically, anything that happens in the cell with molecular processes is driven by some other sorts of proteins and or enzymes. Also, this is figure 18-19. This is figure 18-17, and basically we're trying to understand through this image what sort of regulation enables M-Cdk, the mitotic CDK, to drive G2 to M. We've already heard some about this story, which is including CDC25, which is a phosphatase that activates M-Cdk by removing inhibitory phosphate or phosphates. And we're seeing another part of the story here, which is that once we get some active M-Cdks, they actually will feed back and activate more CDC25 phosphatases, which then this provides a positive feedback loop, because more of the CDC25 phosphatases we have, the more of the inactive M-Cdks that can be converted into active M-Cdks. So let's talk some more about what M-Cdk actually does. So indirectly, it leads to the formation of the mitotic spindle. And the main event that takes place is that M-Cdk is going to activate another protein complex, which is called condensins. And you'll see some more about that later. Here, what I want you to know is that a requirement for forming the mitotic spindle is that chromatin, which is made up of DNA and proteins, which is a loosely packed kind of spaghetti structure of DNA and proteins. It is packed, but not super tightly. Then it gets condensed into a more compact structure called chromosomes. So chromosomes are the compact structure, and they would look like what you are used to maybe seeing them look like this. So those are our chromosomes, but chromatin is more like this kind of a thing. And so it's no coincidence that when we say the chromosomes are condensed into this compact structure, that condensins are involved with that. This is figure 18-18. So during the M phase, there are two main protein complexes that are going to help the chromosome to get into the proper state so that it can be divided up correctly. And those are shown here, one of which is the condensins, the other of which is the cohesins. M-Cdk will phosphorylate and activate the condensin complexes. Condensin complexes are shown up here, these blue circles, and so basically they take the DNA and make some loops in it and start to make this kind of star-shaped structure. And basically as all of these stars stack on top of each other like this, instead of just a whole big mess of DNA, we start to get an ordered structure which starts to look like this, where we have one chromosome here. And then we have its duplicated chromosome right here. And so the duplicated chromosomes, notice that they're like peculiarly very close together. That's not an accident. That's due to cohesin rings, which those are a second protein complex shown here in green that will run the entire length of the chromosomes and hold them together very tightly up until the end of metaphase. So you should know the difference between these two different protein complexes. The condensins, which I've drawn in over here, run the length of each chromosome, and then the cohesins run between the two chromosomes. The cohesins, again, are going to be used to hold the sister chromatids together for as long as they're held together, and we'll see that in anaphase, that's when the cohesin proteins are dissolved, and that means the two sister chromatids can disassociate from each other.